Let's start with our normal review of the warm-up here. Okay, here we go. Again, we have been talking about the brain, and we're actually going to talk a little bit more about the CNS. Actually, we'll spend most of today still kind of in the CNS. Parkinson's disease, we talked about being the degeneration of the substantia nigra, which is in the midbrain. Lentiform and caudate, and mainly motor functioning. And then where do we find the lentiform and caudate nuclei? If you were going to look for them, where are they in the brain?
Aren't they like in the deep cerebral hemispheres?
Exactly. These are the deeper pockets of gray matter in the cerebral hemispheres, the nuclei. And what part of the brain has axons that connect up into these, we call the basal nuclei, the lentiform and caudate here?
I want to say substantia nigra.
Yeah, exactly.
Okay.
That was the whole point about Parkinson's, was the degeneration of the substantia nigra, which then causes a dysregulation of these areas that are responsible for movement. So you want to make sure you kind of remember how everything connects and how that all works. 4 functions of the hypothalamus. Most people did pretty well on this. Again, it's doing all sorts of stuff. It's the main arbiter between connecting the nervous system and the endocrine system through the pituitary. It's doing all sorts of autonomic control, like respiratory control, cardiovascular control, things like temperature, different drives like hunger and thirst, and all sorts of emotional stuff, rage and pleasure and things like that. Sex drive. Most people got this. Coughing, sneezing, vomiting are all the medulla oblongata, the kind of real core, kind of keep your airways clear and expel poisons. Amygdala, again, cerebrum. It's another one of these deeper gray matters. It's the sitting at the tail of the caudate nucleus. So the amygdala is another pocket of deep gray matter in the cerebral hemisphere, but it's more involved with emotion instead of motor. So any other questions about anything that we've covered up to this point? All right, so we're going to continue on. We're almost done with kind of our different regions of the brain. In fact, the only part of the brain we still haven't really talked about explicitly is the cerebellum hanging off of the brainstem. So we'll do that. And then the rest of today is going to be more around either systems, different regions of the brain that work together for a common function but aren't localized in any one particular place. Or also we'll talk about even these more kind of integration kind of topics like memory and learning and sleep and things like that, where you kind of step back and look at things, and there's interesting things to say, but definitely not talking about any particular region anymore. All right, whiteboard. All right, so just if we can finish our talking about the different regions. Let me wait a second here. So do you see my whiteboard right now?
Yes.
Okay, good. So here we had kind of the brain. It was like the midbrain. There's the pons and the medulla. Going down to the spinal cord. Hanging off of the back of the brainstem, kind of connected to the pons by these things called the peduncles, we have the cerebellum. This is actually kind of right up against there. And the cerebellum has connections to all of the motor parts of the brain, the things that are kind of controlling your body. And it also has connections to all of the sensory stuff that is giving information about what is the current position of your body. We're going to talk about more when we do the sensory stuff, like the proprioception. What are the angles of my joints? What's the tension on my muscles? So it's got information both about what you want your body to do and what your body is actually doing. And it kind of helps close the loop to make sure that, people talk about, smooth coordinated movement. Like if you have damage to your cerebellum, you're not going to have paralysis or anything, but instead you're going to have trouble putting your body where you want it to be in space. So like here, let me go back into, well, that's not what I meant to do. If I have my hand and I am trying to grab this stapler, I need to accelerate my hand, go towards it, and then as I get to it, slow down and stop exactly when it's where the stapler is so I can grab it. That's a complicated control problem. To take this big wonky mechanical system with these moments of inertia and all these different levers and different muscles that make it do all these different things, and I want to accelerate it exactly to the place and space and get there as fast as I can, but then decelerate it and stop it and make it totally still. When you have damage to your cerebellum, you have problems more like where you try to reach for something, but you go too far, you overshoot. And then it's like you try to overcorrect and you come back, but you come back too far and you just kind of oscillate around where you want your hand to be rather than being able to just put it where you want it to be. So like damage to the cerebellum is more about that kind of stuff rather than you're paralyzed. So, there are people who give the cerebellum other roles, but I would say, like first approximation, it's primarily about kind of coordinating your motor movements, kind of closing the loop between what your brain is telling your body to do and the sensory information coming back that is telling your brain what your body has actually done and if you have accomplished your desired outcome. Okie dokie. Let's see. So now let us continue. Wait, where? I lost my squirrel. There he is. All right, so now what I want to do is talk about things that are not particularly localized in a particular part of the brain. So everything we've talked about so far is in a particular part of the brain. What we're going to do now is talk about a few things like we call systems, like the limbic system. These are a variety of regions of the brain that are all involved in emotional processing. And they're interconnected with each other, but they're separate regions. And we've talked about most of them at this point already. What are important regions that process emotions in the brain?
The amygdala.
The amygdala. The amygdala, which we talked about, is primarily seems to be around anxiety and fear. What are other regions that are important?
Hypothalamus.
Hypothalamus, totally. And yet there's more.
The hippocampus.
Hippocampus. Again, the hippocampus, important for emotions. We'll also get deeper into the hippocampus later this morning, talking about its role in memory consolidation. And more. We talked about the cingulate gyrus. That gyrus kind of on the center of the brain. We talked about the insula. Again, there's kind of this fascinating thing about the insula. Insula is more around, or has a lot to do with kind of your sense of your body. And even this whole word, feelings. Right? Feelings are kind of what you're feeling emotionally, but feels also this idea of touching, or what does it feel like? This feels kind of soft, or this, and at least to some degree, for some emotions, there are ideas that your body reacts to something, and the feeling is more of something your heart is speeding up and you're getting all sweaty and you're like, whoa, I'm feeling like scared and uncomfortable. But it's not like you got scared and uncomfortable and then the body reacted. It's more like the body reacted and the feeling is kind of something about what is it like to be in my body in this particular state. So there are obviously much more subtle feelings that don't move along with that. Like, I am like slightly jealous that this person has got this job than I didn't when I got passed over. So it's obviously more complicated than just that, but that part does seem to be folded into some aspects of emotion, which is interesting. There's parts of the thalamus that are in here. So there's all these different regions, and there's interconnections, like the interconnections between the hypothalamus and the hippocampus called the fornix, these little axonal tracts. And interestingly enough, we can say these different regions of the brain are important in emotions. When there's damage to these regions, there's problems with emotions, but exactly how emotions are represented in the brain is still something we don't know. That was something that was actually a little surprising for me when I started doing my research. I knew that these regions are important, and I kind of just assumed that we had more of an idea of what does it actually mean at a neural level to be happy or sad or something. And there's been a number of studies trying to figure that out, and we don't have any idea. It's definitely not a particular region. Like, I talked about amygdala being more about fear and anxiety, but you can't take that and extend it at all. There's not like a happy region or a sad region or this or that. It likely has to do with patterns of activity across different areas, but it's at some scale or subtlety that we have not been able to actually figure out. So how exactly emotions are represented neurologically is still kind of mysterious, right? We can take a electrode and make you flick your hand, because we know what part of the brain is controlling the muscles that control your hand, but we don't know so much about what part of the brain makes you feel different things, or how the brain accomplishes that. So limbic system, just a variety of different regions that are particularly important for emotions. Right, in this, I mean, I can just, emotional regulation, this is kind of what I've been looking at, this idea of these interchanges or exchanges between, the more prefrontal areas, executive areas, and these more limbic areas. Like, how much do you have an ability to consciously modulate what's going on in these areas? Like, this is bad, but maybe I can put more of a positive spin on it, and we call like reappraisal. Like, okay, if I look at it slightly different, it's not as bad, or this or that. So there's a whole level of back and forth between these more cognitive areas and these more emotional areas, and the amount of connectivity even between those and different people is different. Like, you can have, it's not always just purely about willpower. Like, why can't you just like kind of see this like and not be quite as sad about it or something? It really seems some people have more or less kind of what we call functional connectivity between different regions of their brain. So some, and it can change through different kinds of activity and training, even like how much activity in one part of the brain can actually influence or modulate the activity in other parts of the brain. So it's not just you're not trying hard enough. Some people just have more or less of an ability to modulate activity in different areas, it seems. Reticular activating system. So this is another thing where we think of a system rather than a particular region. This tends to be more kind of in the pons and the medulla, but it's a variety of different interconnected regions and neurons that then send activity up into the cerebrum. And this is involved with what we call arousal. And arousal meaning like not being asleep, right? Not like kind of sexual arousal or anything, just like, arousal of like being awake. And you need this activity to be awake, right? It's part of the switch. There's lots of things. We're not going to get into all the details. We'll talk more about sleep, but this activity is necessary to be awake. Like, if you have damage to the reticular activating system, someone will go into a coma. Like, what's the difference between a coma and being asleep?
Being able to wake up.
Yeah, exactly. Coma is non-arousable, right? If I'm like, if you were in class and fell asleep and I saw and then I kicked you, you go, whoa, and you wake up. If you're in a coma and I kicked you, you would just still keep lying there. That's the difference. A coma, you can't wake somebody up from a coma. There's still have, we'll talk about, their EEG still has, brain waves, usually kind of the really slow, like delta waves, like they're in a really deep sleep, but you can't shake them out of it. You can't wake them up. So reticular activating system, it's worth knowing about, because particularly if there's damage to that, that's going to cause some major problems with not being able to regain consciousness. Okay. What else do I want to say? So speaking of EEG, this is might as well just introduce the idea of EEG. And actually, let me get my little. This makes sense to just do with my little typing tool. Electro Encephalogram. Again, electro encephalogram. So it's just electrical activity of the, like encephalitis is when you have inflammation of your hemispheres. This just means of the brain hemispheres, and gram is like you get a picture of it. As opposed to like, right, an EKG or an EC, electrocardiogram, electrical activity of your heart, or electromyogram, electrical activities of your muscles. An electroencephalogram, you basically take this person and you put a whole bunch of little electrodes on their scalp. Usually there's kind of a ground electrode on their ear, and you can just take a look. What is the electrical activity on the scalp that is ultimately being generated by electrical activity of their brain? So their brain is inside their skull, obviously. But their brain is got all sorts of stuff going on, like the sum total of whatever electrical activity is happening on the cortex, and then kind of distorted and warped through all of the tissue and fat and bone and of your skull. Whatever all that is all together is the EEG. So it's not going to tell you anything about any particular neuron or anything like that. But why it's useful is there are typical patterns of EEGs that we measure that are typical for different kinds of states of, like if you're awake versus asleep. And then also ones that are not typical that are going to be indicative of some kind of problems. So we are going to talk about some of the main EEG patterns and when you find them, and that will also help us talk about sleep, because a lot of the ways we talk about the different stages of sleep are by describing what kind of EEG patterns do we see in the different phases of sleep. So let's do this then. So again, these are different types of EEG patterns. We'll start with alpha waves. So alpha waves, these are relatively slow. And I should mention, they're not that pretty. But people, especially people who are good at measuring these things, they look at this, they typically tend to like figure out, okay, how often there is some basic pattern where it's going up and down. What is the frequency of the main kind of oscillation? For alpha waves, it's usually somewhere around 8 to 13 hertz. And again, what is hertz? What does hertz mean?
Frequency.
Cycles per second.
It's the cycles per second. So it's kind of going up and down somewhere around, around 10 times up and down per second. And this is what we find when someone is awake and relaxed. So someone's just kind of chilling, but not asleep. We'll see as you start falling asleep, things change. So this is awake and relaxed. If I all of a sudden tell you, like, start with 100 and subtract 7, and then subtract 7 from that and subtract 7 from that, it's going to change. Or even if I just clap or I snap or something, it's going to change because you're going to pay attention. Like, what's that? I need to focus. So in that case, when you are kind of concentrating and alert and trying to figure something out, it goes into these faster, smaller things we call beta waves. And the B is Greek beta. This is like, I have 14 to 25 hertz. You know, and again, it's kind of going up about twice as fast as this. And this is, again, alert and concentrating. You know, and it's really easy to see. We had a student doing, who actually was an EEG tech, who decided to do her independent research project looking at EEGs under different conditions. So we had these people hooked up in the lab with their EEG electrodes on, and it was just kind of fun. You could see as they're relaxed, it's kind of alpha waves, and all you had to do was like snap. As soon as you snapped, it went like, you know, you could see it. And then if after a while it kind of goes back because there's anything going on anymore, they're going back into that relaxed alpha. So just kind of, it's something, it's not like theoretical. If you have somebody hooked up with those electrodes on their scalp, you just let them relax, it's going to start moving towards this. When you have them like have to concentrate, it goes to this. So just kind of putting that out there. So these are ones that are typical when you're awake, either relaxed or alert and concentrating. The next two I'm going to do are much slower, and these are ones that are only found when you're asleep. If you see them when someone's awake as an adult, it's usually a sign there's some major problem. So theta waves. This is 3.5 to 7.5 hertz. You know, so much slower, much bigger. You know, if you're a kid, these can be normal. Like the EEGs, as you go through different ages, change what's typical, right? Because I mentioned that up until your early 20s, the brain is like rewiring and growing. So when you get like a textbook with normal EEG patterns, it's by age. So something that might be fine for a kid, if this shows up in a kid, that's nothing to worry about. If this shows up in a awake adult, it is something to worry about. We're going to see this in a few moments when we talk about what happens as you get down into the deeper parts of slow wave sleep. And then the slowest waves we have are called delta waves. You know, 3 to 4 hertz or less. You know, these are what we're going to find in the deepest parts of sleep, or if someone's like in a coma as well. So all right, so these are kind of classic EEG patterns. So what do I want to do now? So this is as good as time as any to kind of then introduce idea of sleep. So let's talk about sleep. So sleep, obviously we need to sleep. If you don't sleep, all sorts of things go wrong. You start hallucinating. You start people who've actually, like, for different challenges have tried to like not sleep for a really, really, really long time. Some of them have ended up kind of messing themselves up pretty bad, and just actually having all sorts of psychological problems and things. Like, sleep is useful for lots of reasons. I can talk about a few things that we know, lots of things we don't know. We do know that sleep seems to help consolidate memories. Like, if you are learning stuff, if you have a person who is studying and learning new stuff and they get a good night's sleep, they are much more likely to retain that material better than somebody who does not get enough sleep. It's one of the reasons I encourage, even though I know when you're in college particularly, and actually even now when there's so much weird crap happening, it's sometimes hard to find enough time to sleep. You want to do your best to get enough sleep for yourself, because if you can study and study and study, and if you don't sleep, you're going to be not as good at retaining what you're trying to study. During sleep also seems to be a place when we get to kind of clean out the environment around the neurons in your brain. This is something that's kind of more recent. This idea of what they call the lymphatic system. We're going to talk about the lymphatic system a little later, kind of this way that you kind of drain leaked fluid out from between the cells in your body. And the dogma has always been like, the lymphatic system is everywhere except in the CNS, and in bones. And it's like, what they've found more recently is there's something similar to it in the brain, where during sleep, there are these kind of changes in pressures, and there's kind of bulk flow of fluid that kind of goes through between the cells that helps kind of clean things out and refresh the extracellular environments around your neurons. So part of sleep seems to be some kind of physical kind of maintenance of just the brain tissue. There's also, like I said, more kind of things like helping consolidate memories. You've got all this information coming in constantly. What do you let go of? What do you hold on to? So sleep. Let's talk about, there's two main types of sleep. So when you sleep, you go to bed at night, you kind of go back and forth between two really different states of both physical, both in terms of EEG, what the brain looks like, and physical, even what your body is doing. So when you start sleeping, we start what's kind of slow wave, also called non-REM. So as you start going to sleep, you're going to probably be kind of like an alpha wave, right? Because you're relaxed. But then you start getting these bursts called sleep spindles. This is where we know that someone is falling asleep and not just relaxed. So these sleep spindles start happening. This seems to be related to these like potassium currents actually that start coming in and creating this different state in the neurons in your brain than they normally experience when they're awake. And then we start just getting more into the slower stuff. You get into theta, and the deepest parts would be like delta waves. So this is a slow wave sleep. And it can go just down, 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 but it can also, kind of go down and back up and down, and, kind of, if this is like, delta, and this is, so it's not necessarily you go down and you come back up. It can kind of hang out and kind of go up and down a bit. And in this slow wave sleep, your heart slows down, your temperature goes down. Again, your EEG is super slow, and you're not really moving because your brain isn't doing anything. Your brain is basically like in this super slow holding pattern. And so you're lying there because there's no activity in your brain to operate things. There is slight movement, like about every 15 minutes. Move around every 15 minutes. There's some little subroutine that turns on, so you don't end up getting bed sores, right? If you're just lying on your body and you don't move at all, you'll block circulation. And right, you've probably noticed that if you sleep on something wrong, all of a sudden, whoa, it's not working right. Or if someone's in a hospital, you have to shift them around so they don't get bed sores, so they don't start losing circulation to parts of their skin and start getting tissue damage. So you do move a little bit, to keep from getting bed sores, basically. But in general, you're just kind of just lying there, limp. There can be some weird stuff that goes on here. You can get sleepwalking, night terrors. Sleepwalking is a weird thing. It's like your body, even though your brain is in general not on, these kind of like things turn on where you get out of bed and you can walk. There's even sleep eating, where people will walk to the refrigerator or the cabinet and get food and like shovel it in their face and then walk back to bed. Like there's been cases of people who aren't losing weight and they ultimately find out that they're doing this unconscious eating in the middle of the night. So dreaming we're going to see is typically in a different part of sleep. But so this is a slow wave sleep. So when you first start going to sleep, this is kind of usually where you start. So, and they call these different stages, sometimes stage 1, 2, 3, 4, or 1, 2, 3. It doesn't matter. But basically, when you start your sleep, this might be the first, if this is hours here. When you first start sleeping, this is kind of this slow wave sleep. And then there's this just switch that goes, and you go into this really different part of sleep called REM. It's probably not that long. Let's make it a little shorter. So REM, this is rapid eye movement sleep. So in REM, it's really different from that slow wave sleep. EEG looks like awake. You get like beta waves, etc. So the brain is really active. Right, which could be a problem. If your brain is really active, like you're awake and doing stuff, and your motor cortex was controlling your body, you'd be running around and doing stuff. So there is this interesting thing where the body, the muscles, skeletal muscles are paralyzed. Except, for little eye muscles, and some inner ear muscles or middle ear muscles. So the reason it gets the name rapid eye movement is that's some of the only muscles that are actually still connected to your kind of motor control. So as they've actually showed if you're in a dream and you're looking around, your eyes in your head as you're lying in bed are actually following, moving, and doing what your eyes are doing in your dream. So rapid eye movement gets its name because your eyes are wiggling around because they're still under the control of your motor system, but everything else is paralyzed. There are these sleep disorders where that sleep paralysis gets doesn't work right, and then you get weird things where people like act out their dreams and they'll like leap out of bed and leap out a window. We had one patient where that when I was taking a class where he'd wake up in the middle of the night and would like start like hitting his wife who was lying next to him because he was having a dream where he's actually protecting his wife from an assailant and he's wailing on this guy, but actually in reality he's lying in bed and he's actually wailing on his wife. So there's things like weird stuff like that if this paralysis isn't working properly. So EEG is like awake, as opposed to, remember, in slow wave, it's like delta waves. It's like super slow. Your heart rate and blood pressure go up. Your body temperature goes up. Right? Sometimes they call this paradoxical sleep. Because your brain is active and your body is activating. So it's a very different state than you experience in that slow wave sleep. And then after you spend some time in REM, you go back into slow wave. So actually I kind of drew this, so I'm put myself in a box here. But basically you're kind of going up and down, you go into REM, then you kind of go back into slow wave, and then you go back into REM, then you go back into slow wave, and you go back into REM, back into slow wave. Usually somewhere around 3 times or so during the course of the night, you alternate between these different forms of sleep. And the amount of time you spend in the different phases and how deep you go into slow wave varies through the night. Usually the deepest parts of the slow wave are in the beginning of the night. The amount of REM depends on how much you've been sleeping. Like if you haven't been getting enough REM, you get into what's called Rem Rebound, where you have extended periods of REM. So the amount of the different kinds of sleep and how deep you go depends on how far into the night you are, what previous amount of sleep you've been getting already, things like that. So it can vary. REM is also where the majority of dreams happen. So most dreams happen in REM. And I should mention, even though in our language we think of dream as like a cool thing, like, oh, like my dream house or my dream job, or oh my God, he's so dreamy, or whatever. Most dreams are actually not that pleasant. I have here, what is it? The study. Where is it? Oh, here it is. Calvin Hall. So this researcher cataloged 10,000 dreams and basically found like over 2/3 of these dreams, when they're just like having people report random dreams, over 2/3 were sad, apprehensive, or angry. Less than 20%, like 18%, were happy or exciting. So dreams seem to be places where people like kind of work out like problems or anxieties or things like that. So if you find that you're not all, everybody else must be having happy dreams, how come my dreams are like my dreams kind of suck? It's like, no, that's normal. Dreams are usually kind of weird and freaky, or anxious, or again the classic thing like, oh my God, I'm teaching and I'm in class and I forgot to put on my clothes or something. So dreams. Other thing about REM, just physiologically, this is where men tend to have spontaneous erections. Whoops, I spelled that correct. It seems to be kind of part of the way the system kind of keeps itself functioning, and it's clinically a useful thing, because if some guy comes in with erectile dysfunction and they're trying to figure out, is there something physiological happening where there's something keeping the penis from engorging with blood and getting erect, or is this more of a psychological thing? Because often the erectile dysfunction can be from, and we'll talk about it, it's like a parasympathetic reflex, and can not work if somebody is too kind of anxious or tense. So if you want to see, is the basic just physiology working, what they do sometimes is they'll put a little band of paper around the base of the penis before someone goes to bed. And assuming that the basic physiology is functioning properly, then that little band of paper will be broken in the morning, because the penis will have swollen during the night because of this, and will have broken the little strip of paper. So it's kind of a interesting little thing, a little clinical aside. So sleep. Definitely, like I mentioned, you want to make sure you get enough sleep. If you don't get enough sleep, you don't tend to retain stuff as well. Your memories don't set as well. Just your mood is not as good. There's lots of things that seem to happen during sleep that are useful in terms of being at optimal, being at your optimal kind of functioning. I wonder if I have, hold on one second here. I am, oh, here. So this, this is just from some stupid, oh, come on. Here. This is just from some random website, but it looks about right here. This idea of what these kind of hypnograms look like. Like when people have sleep disorders or they're trying to figure out what's going on, they'll actually put them into a sleep lab, and they'll actually have them hooked up with EEG electrodes, and they'll have them sleep, at the lab. And again, this is kind of showing this idea of what I was drawing, stage, and this is, hours through the night, starting with kind of alpha waves, but then going into sleep spindles, then these slow waves, and then stage 4 are like these slowest waves, the delta waves, but then you come back up. Then you got REM. Everything is like your brain waves are fast, your body's getting hot, and your heart's speeding up. And then, slowing down again. Then back up and REM, and then slowing down and back up REM. And again, these final stages, you're not going as deep into the slow wave, and finally, like, you're awake. So that is, and I should also mention, when we're talking about sleep, some of the stuff about this idea of, you're supposed to go to sleep and then wake up in one big block is, that seems to be kind of more modern, like, eat 3 meals a day, a breakfast, lunch, and dinner, and you use a fork and a spoon and a knife. These are just kind of cultural things that kind of come into vogue. But there's been times, especially like when people didn't have like, artificial lights and stuff to kind of create this thing, people would have like 2 periods of sleep. They'd sleep, then they'd wake up and they'd do stuff and sleep more. So this idea that there's 1 block of sleep versus 2 blocks of sleep is a little artificial. How much people need to sleep is really different. When they put people in these caves where there is no light to tell them when is day and night, you have some people who tend to have less sleep that they tend to do, other people that have more sleep than they tend to do. Again, for the purpose of this class, we don't have enough time to kind of go into all of the different styles and things. And even things like naps, depending on your kind of sleep style or kind of how your body works with sleep, depending on how much you've been sleeping and what time of day it is, a nap can be amazing or a nap can be actually counterproductive. For some people, naps, like a 15-20 minute nap, can totally create a new lease on life for the day. Other people, it just leaves them stranded in this place where their brain is in a fog. So, what is right for somebody really depends on that person and other things like how much they've been sleeping up to that moment, and also even the time of day, because it depends on the circadian rhythm, like stuff that's going on in their body. So yeah, sleep gets really complicated. It's fascinating, but it's complicated. So sleep. Let's now shift gears and we'll talk about memory. Memories. So memory is another one of these things that is really fascinating, and we're learning more and more about. And we'll talk at kind of a superficial level. Memory is, obviously a way of kind of recalling something, bringing back some moment, but it seems it's not like a particular place in your brain where a memory is stored, right? You can actually have a memory of the time you were running around doing something with your friends, and then you have damage to your visual cortex. And the visual part of that memory will be gone, but the rest of the memory will still be there, right? So it seems to be at some level this kind of reactivating or recreating kind of a pattern of activity that happened at a certain moment, but in kind of a less intense or real way. So it's, right, you can actually have problems like with PTSD and stuff where people can't distinguish that this is just a memory. It just feels real again, which can be really messed up. But normally, you know that it's a memory and it's not actually happening to me right now. It's like something that happened. So some of the understanding of memory has been really fascinating as we've learned more. Like this idea, let's talk about short-term memory versus long-term memory. So short-term memory is like right now, when you're first thinking of something or hearing something or connecting things, it's kind of this more way you can juggle things around and hold things and manipulate things. And it seems to be more of kind of in electrical patterns in your brain. Long-term memory seems to be more of a physical, like kind of reweighting of synapses and rewiring things. Like if you give somebody electroconvulsive therapy, ECT, that's electroshock, which is still used for some people. It's critical. Like it can be really useful for like resolving of intractable depression. If somebody is really depressed and you're worried that they're going to commit suicide, and there's a good chance, even if they commit suicide before the drugs kick in with the antidepressant drugs, this can be a way to instantly make somebody feel less suicidal. If you give somebody electroconvulsive therapy, put this electric current across their brain, everything that they experienced like for the last 15 minutes is gone. But everything from an hour beyond is still fine, right? Just to give you an idea, like short-term memories and some more temporary electrical patterns in your brain that's going to get wiped out if you put a big current through the brain. Whereas the long-term memory is more permanent. It's got, again, it seems to actually be, we're going to talk about experiments that like block protein synthesis will block the formations of long-term memory. You need to actually kind of create structures and reweight synapses and stuff to make long-term memory. The process of moving things from short-term into long-term memory, they give a name called consolidation. So something is in short-term memory, you can move it into long-term memory. Things we do know about consolidation is the hippocampus is critical. People who have damage, bilateral hippocampal damage, don't form long-term memories. Like if anybody's ever read The Man Who Mistook His Wife for a Hat, like Oliver Sacks, he died a few years ago. He was a neurologist who wrote so many amazing books. Like if you're interested in neurology and the breadth of human functioning, he's written so many amazing books. Like pretty much anything he's ever written is worth reading. Whether it's about all sorts of different neurological disorders or about the way music is processed or hallucinations or anything, he's good. But he describes this one guy he calls the Lost Mariner, in one of his books. it was like actually through chronic alcoholism, he actually ended up with his hippocampus is no, damaged. And every time he met somebody, he would still have his autobiographical knowledge of who he was up to where that damage had happened. But even though he was now, like, I think in his 50s or something, he still kind of thought of himself as a 20-something-year-old because none of the new memories that he had made going through life were actually being converted into long-term memory. If he met you, he'd be super friendly, he'd seem totally normal, except if he met you again like an hour later, it would be like if he met you for the very first time, because none of those memories were getting converted into long-term memory. It's like the dude in Memento. If anybody saw that Chris Nolan movie, that's an example of, that was kind of a real life version of that dude in the movie, where any new memories just kind of evaporated after like a half an hour. So consolidation. So things that have been really interesting now is people realizing that when you store your memories, it's not like you put them in a file cabinet and you peek in the file cabinet to see what that memory was, if you're remembering it. If you want to use an old memory, you actually have to bring it back into short-term memory. So you bring things out of long-term memory into short-term memory where you can kind of manipulate them and use them. And then they get put back in this thing called what they call now reconsolidation. So this brings up some really fascinating issues. One thing about reconsolidation is you can contaminate the memory while it's back in short-term memory. So every time you remember something, there's a danger that that memory is actually getting mutated. They did this really interesting study where they had people recall their times at Disneyland when they were a kid. But then the researchers were kind of sneaky and would kind of suggest these, experiences with people in the Bugs Bunny suits and things like that. And by the end of this, these interviews with the researchers, people would have these vivid memories of being at Disneyland and having some particular experience with somebody in a Bugs Bunny suit, and, they knew that that couldn't be real because Bugs Bunny is a Warner Brothers character, would never be at Disneyland. Disneyland would be Mickey Mouse or Donald Duck or something. So they knew that it was a fake memory that happened because they brought in their real memories and then they got contaminated and then got back here. And how likely it is that the memory gets changed, can depend on different things, like kind of your just kind of sympathetic activation and things like that, how intense things are in the moment. But it's possible that, memories, like this idea of your memories just being this constant thing it's not really true. It's a little scary to think about, but your memories are kind of slowly like morphing in the past. You might have even noticed sometimes, like, you go back and you see some picture or you read something like, wait a second, that must be that because that's what it looks like in the picture, but it's not exactly what I remember. Or if you've ever been in a trial or something and been on a jury, it's just bizarre to see how different people's memories and experiences are of the exact same experience. So other things that are fascinating about this reconsolidation is people have done studies where they give a drug that blocks. So these are studies like in rats and things. They'll give a drug that blocks protein synthesis. And when you do that, it blocks reconsolidation. So you can give a rat a drug that will block the ability to convert, short-term into long-term memory. And then they did this study where they took these rats, and they had two mazes. Right, so both rats learned maze A and maze B. And by the end of the training, both rats could do both mazes really well. And then what they did was they gave one of the groups of rats this drug that blocks reconsolidation by blocking protein synthesis. And then they had both rats do maze A, which means they have to remember how to do it so they can do it. And then when they were done with the experiment, they then checked, and the rat who was just doing the maze still totally remembered both mazes. But the rat who was having to remember maze A while he was having his reconsolidation blocked, he still remembered maze B. It didn't affect this, but he now did not have the ability to do maze A anymore. So it basically erased the memory. So the implications are kind of trippy. It's kind of like, again, when I think about movies, that like Eternal Sunshine of the Spotless Mind, like theoretically, it is possible to go in and erase memories by having someone bring that memory back into short-term memory, but then blocking reconsolidation so they can't put it back in. Ethically, there's all sorts of issues around, like, is that a good thing? It could be great to like get rid of traumatic memories, but then how much of our personality and who we are is based upon our memories? And what happens if you go in and you get rid of stuff that you didn't mean to, or who knows? It's tricky, but it is a very trippy thing to think that, that's actually a conversation people are having because the technology is kind of close. Should also mention here, just because the hippocampus, I've mentioned hippocampus is critical. And again, hippocampus was also essential part of your limbic system. How strongly things are shifted from short-term into long-term is modulated by kind of the emotional valence of things, right? If something like, the thing I often say, like, in 20 years from now, you'll probably even forget who taught physiology class. It's like, okay, I'm a nurse, I had to have taken physiology, whatever. But like, what are the details? Definitely the details of this day, you're not going to remember at all. Like what, but if I, this was the day, I can't take it. The pandemic is too much, and I've got like my little rabbit squirrels here. And you watch me getting ripped apart and blood splattering across my little like, webcam here, and then it's be in the news tomorrow and it's tragic. You're never going to forget this moment of watching your teacher get ripped apart by squirrels on the screen, right? Because you've got this really intense emotional component to the memory, and that's going to create a much stronger consolidation there. So that's worth, yeah. So memory is fascinating. While we're talking about memory and learning, we can also talk about two different versions of this. Explicit, also called declarative. versus implicit, also called procedural. So this can refer to learning or memories, because again, what is learning? What are memories? Memory are stuff you remember. Learning is the stuff that you remember. Declarative or explicit. This is declarative, meaning I can say what I know. Like I can say that the cerebellum is responsible for smooth motor movement. You know, if you ask me, what do I know? I can tell you using my words. That's why it's also called explicit. I can explicitly say what it is that I know. Implicit or procedural, this is stuff where your nervous system is actually noticing things. You are learning things that actually influence your decision making, but you don't actually explicitly know what you know. So like when I think about like intuition and things like that, where it's like you get a sense of like, this is the right thing or not the right thing. I'm not exactly sure, but I got a hunch here. It's often because you've got like information that you don't just have explicit access to. Like examples of like in studies, the way they would do this, they would like show you pictures and say, you know, this picture is a yes. You know, this picture, that's a no. You know, there's a training. You know, this picture is a yes. This picture is a no. This picture is a yes. You know, this picture is a no. And you're like, whatever. And by the end, you're like, I have no idea. It all just looks like random dots to me. But then they say, okay, here you go. Is this a picture a yes or a no? And you're like, I don't know. I don't know what this rule is. And you say, just guess. So people guess. But it turns out, you know, there is some rule that they never noticed. Like the rule might be like, you know, it's a yes if there's like a higher density of dots on one side, and it's a no if it's a higher density of dots on the other side. But they never actually explicitly figured it out. But implicitly, there was something in their nervous system that was learning that did notice it. And now they have to choose like, yes, this picture is a yes or a no. They feel like they are randomly guessing, but in fact that they're guessing above chance, meaning that they did learn something, they did notice something, it's affecting their decisions, but they don't even know it. They don't even know what they know. So that's this idea of implicit or procedural learning. And it's pretty clear that kind of the neural substrates for these two kinds of learning are different. Like that guy I was talking about in that Oliver Sacks book who couldn't remember you if he met you 1 hour from now. It turns out they did this interesting experiment where, you know, he was living in a hospital because he couldn't live on his own, and every morning they would say, you can go like with, you know, Dr. A or Dr. B. And so, you know, because he had, you know, to do some test, and every morning he would, you know, whenever he saw them, it would be like it was the first time he ever saw them. He never knew who they were because, you know, he couldn't make those long-term memories. But they had it so every day Dr. A would be kind of mean and Dr. B would be really nice. And eventually he would, you know, even though in his explicit memory they were two random new people he'd never seen before, I don't know, let's pick B. I'll go with B. He would start, you know, so something in his learning, he started realizing this person is nicer or safer than the other person. And they actually, part of the control they made, the meaner doctor be like this, like kind of cuter woman that you probably would have picked more just on that. So there is this implicit learning of, in that movie, Memento, he doesn't exhibit this, but in the real cases where they've looked at it, it seems like a very real thing. Like you can preserve implicit learning even if explicit learning is not functioning. So there's a different neural substrate. Right. So again, explicit, also called declarative, because if I knew, like, I'm picking this doc B because I know that they're nicer to me and that other doc is a jerk. But implicit is, I don't even know why, but I'm going with doc B just because it just seems like the right thing. I don't know. Right? You don't know why you're picking doc B, but there's something in your nervous system that has learned something just through the process of going through things. All right, what else do I want to do? So any questions about any of that stuff?
So you kind of explained or described short term as temporary electrical patterns in the brain, and long term was what?
Kind of more permanent physical changes, like things where there are different weights, like weighting of synapses, actually making or changing the number of channels that get activated, or how many dendrites are connecting to axons or things like that. So there's actual kind of rewiring going on. So an actual physical change in the circuitry rather than just kind of temporary electrical patterns that are changing.
Thank you.
So what we'll do, just to finish off the CNS, I'm going to talk very briefly about the spinal cord, because the spinal cord is part of the CNS and I don't want it to feel like left out, but we're not going to focus on it a bunch, but I should mention it. So spinal cord, a lot of the spinal cord is what you kind of think. It's kind of messages coming down and coming up to communicate, connect the brain to the rest of the body. But it does have some integration. We talked about the spinal reflexes, right? Like that knee, patellar tendon reflex, withdrawal reflex. Actually, some of the sexual responses like erections and stuff like that are happening in the spinal cord. So there is some integration. A lot of damage to the spinal cord. It's worth kind of understanding the basic idea of kind of some of the main nerves that come out. At the very top, the spinal nerves, we call these spinal nerves, these things that are coming off of the spinal cord. And remember, a nerve is just a collection of axons. These are both kind of motor and sensory axons that go off to connect to your muscles and your sensory receptors all over your body. So when people have damage to their spinal cord, that means that anything below wherever that damage is not going to be able to send the message in or get messages out from the brain. So depending on where the spinal cord has been damaged or cut, you're going to have different kinds of problems. At the very top, it's worth the only nerve you need to know by name for our physiology class. Kind of coming out from the very top of the spinal cord, we have the phrenic nerve. This innervates the diaphragm. The diaphragm, which is the main thing that drives pulmonary ventilation. So if anything happens that stops messages from going from the pons and the medulla to get to the phrenic nerve, you're going to die, unless you get on a ventilator like in minutes. So Spinal Nerve C3 through 35 are the ones that kind of make up this phrenic nerve. The little mnemonic people use is C3 through C5 keep you alive, right? So if you have damage to your spinal cord where it's cut at C2, you are not going to be able to breathe. You have to be on a ventilator to stay alive. If you get below C5, you're not going to die from not being able to breathe. But below that, that's where you're going to get the brachial plexus. You're not going to be able to operate your arms or your legs or anything. So that would be like kind of a quadriplegic. If you get below the thoracic level, that's basically below the brachial plexus. So your arms will still work, but the stuff below your arms won't work, have either motor or sensory. That would be like quadriplegic. Right, so depending on where the spinal cord transections are. And some of that stuff I was talking about in the PNS, like with the Schwann cells and the Wallerian degeneration and all that, where axons can regrow and reconnect, that doesn't work in the CNS. People are spending a lot of energy and time trying to figure out how to have axons in the CNS repair, but it's not so clear how to make that happen. We're going to see there's also implications about spinal cord transections for the autonomic functioning, but we'll get to that when we do autonomic. Other things we can mention here. Just about spinal reflexes, like fascinating things. Like I said, erection is a spinal reflex. So you can actually have kind of a physical, like just physical touch, stimulation of sensory receptors on the penis. So the signals are going to the spinal cord, the signals coming back that results in erection. But since nothing ever actually made it to the brain, the person did not experience anything. They didn't feel anything, even though there was actually a reflex. There was a stimulus response, but the same thing like with knocking on somebody's knee. You might have them kick out, but they'll have no idea that they even got touched on their knee because the signals didn't make it up to the brain. So these spinal reflexes can happen without any issue with the brain knowing what's going on. So spinal cord. I guess that's going to be that. And so with that, we have finally checked off the CNS. So we still have the PNS to cover. The PNS is going to be, motor, which will be relatively straightforward. This is going to be the somatic and the autonomic, sympathetic, parasympathetic, and then the sensory, which is going to be a lot more involved, which we're going to we started already with the general senses. We're going to continue on in lab today with the visual stuff.